SFBBO, promoting sustainability by engaging communities for more than 40 years. Our Birdie Hour speaker series is a free virtual program. On February 20th, 2024, PhD student Trina Chou gave a talk to our community about how neuroscience is used to study hearing in songbirds.
And thank you to everyone who's coming out tonight for the talk. As Kristen said, my name is Trina, and basically I'm excited to talk to you guys today about how we can use neuroscience to study birds. And so I'm sure many of you are here because of our interest and love for birds, but I'm kind of going to give a brief introduction of both the topics I study and then the birds that I can then use to answer my questions of interest. And so I'm really interested specifically in kind of acoustic communication as a general topic, right? So the way that animals are able to communicate with one another acoustically. And so there's lots of scenarios of animals communicating with one another, but at its most basic, you can kind of think of it as two individuals, such as a male, right, singing for a female, right? You hear a lot about songs. And so you can see the male, who's going to be pictured on the left, right, is singing some sort of song that then is going to potentially attract some female. And so in the context of my research, and when I talk about research, I typically think of them as the male as the signaler, right? He is the one who's producing some kind of signal, his song being that signal, and then the female being that receiver. So the receiver is going to be the one who hears the signal and extracts some kind of information from it. And so by using these terms, you can kind of use them across multiple different scenarios, but you're still describing kind of the same general aspects of communication. Now, the cool thing is that animals aren't just, of course, communicating in pure silence. We live in environments, we live in these places that sound very different. And so signals often have to travel through these environments that they live in. And these environments can impact different aspects of their communication. And so as you can kind of guess, right, animals live in a variety of environments. They sound very different from each other. They look very different from each other. And even the same kind of animal can experience two very different things, especially when you think of things like background noise or soundscapes. And so noise can then influence kind of the ability of different species and different individuals to do different things in respect to communication. So it's important to kind of think about the existing soundscapes that animals live in to understand how they're able to communicate in them. And so, for example, birds, right, living in the middle of a very crowded city are experiencing something quite different from birds who are living in kind of an uncrowded, quiet backyard. And these are going to have different kind of implications for the ways that they're able to communicate with one another. And now the cool thing is this doesn't just apply to birds. It can also be true of frogs who often have to deal with waterfalls if they live near ponds versus those that live more in quiet forested areas. Crickets are another great example. And even bats, right? There have been more of this kind of urbanization of bats into these different actual going under highways. And so these different animals, same animals, right? But they deal with very different environments. And so it's kind of interesting to think how this noise is going to shape the way that they communicate with one another. Now, recently, too, a big kind of topic of thinking about noise is actually anthropogenic or human noise. Basically, due to various human activities, the kind of over the past decade or so, the general levels of anthropogenic noise have dramatically increased, and they're projected to also kind of increase in the future. And so noise can influence several aspects of communication. And so it's something we want to think about as, these environments that birds and other species have adapted to kind of deal with are now changing with the addition of human noise. The question is, how are they going to kind of respond to those changes, especially, in the context of this? So there's a couple ways that noise affects communication systems. One of them is that it can affect the way the signal itself travels through the environment. So for example, a bird call that's played both in, a quiet forest versus in the middle of a busy city, right? Those signals are going to travel differently just depending on which environment it's played in. Similarly, it can also affect what kind of sounds the producers or the signalers are making. So for example, if you're trying to talk to someone and it's super noisy in the background, you may adjust your own volume, right, to kind of compensate potentially for that noise. And then finally, too, noise can affect what the receiver is able to hear, right? So if you're on the flip side of that, if you're in a really now noisy, kind of crowded situation, it may be harder for you to pick up what that signal is. And so this kind of last third bit is what most of my research is interested in, and is classically kind of been an understudied aspect of bird communication. So for example, there's been a lot of studies showing that in, recent and increasing levels of anthropogenic noise, birds actually have been shown to shift the frequency of their songs upwards, so singing at higher frequencies, and also sing at louder amplitudes. The two might be correlated due to what's called the Lombard effect. But point being that there's been a lot of research into the signaling side of things and what signalers are doing to kind of compensate for this noise, but not as much research into what the receivers are doing. And so that's kind of where my research takes me. And so giving you the general brief landscape of acoustic communication. And so now I can talk to you about the birds that I use to study this concept switch. So here you see 3 pictures of our birds of interest. We've got our black-capped chickadees, our tufted titmice, and our white-breasted nuthatches. When it comes to kind of hearing and understanding hearing, there's a standard assumption that all birds are kind of hearing things in the same way, or that their hearing systems work in the same way. And then that, their hearing is going to be similarly affected by noise, but that may or may not be true. And so songbirds are great to kind of study this because there's a lot of different species of birds that have different communication systems, and they use communication for different things. And so species may not only communicate, with individuals of their own species, like kind of the male-female thing, but they can also communicate with individuals of other species. So our 3 songbird species live in various urban and rural environments. And the cool thing about them is they make mixed flocks together, which I'll talk about in a little bit. But first I figured I'd play you some calls. So this is where the audio kicks in. So feel free to adjust your volume as needed. But so first I'll play you what a typical chickadee call sounds like. I'm sure many of you are familiar with this sound, even if you don't realize you have been. But so that's the typical chickadee call, or it's a chickadee-dee-dee. And there's different kind of stereotyped notes. And what you see on the screen is what's called a spectrogram. So with time on the x-axis and frequency on the y-axis, it's basically kind of a visual representation of what that call sounds like. And so next I'll play for you a titmouse call. And so you can see it looks a little different, it sounds a little different, it has a little bit more of those higher frequencies in their vocalizations, right? Because these 2 spectrograms are kind of spanning the same Y dimension. And finally, then here's the nuthatch call. I personally really like the nuthatch calls. I don't know why, but I really do. And so these ones, anecdotally, from what I've noticed in the field, tend to be the quietest kind of the 3 species. And they also have way more kind of those lower frequency components. And so, as I mentioned, the cool thing about these birds is that they form these mixed species flocks during the winter and non-breeding season and do this cool predator mobbing behavior together. And so chickadees and titmice in particular will make these alarm vocalizations to warn both, individuals of their own species, as well as the other species, that there's a predator in the area. And then by doing so, then they will collectively get together to harass and mob that predator. And so this is just a video I found off of YouTube of what I believe is a Cooper's hawk being mobbed by chickadees. And so you'll hear the anti-predator response kind of manifest itself in a couple ways. One is going to be through actual physical approaches, or at least kind of chickadees surrounding the area that the hawk is in. But then also importantly, you'll hear a lot of alarm calls. And so, even though we won't be able to see many chickadees in the video, just by the audio alone, you can tell there are many of them. always love watching these mobbing videos. But yeah, so basically kind of to sum all this up, what my research and what our research in Dr. Gall's lab was interested in knowing is basically how this anthropogenic noise is going to affect the behavior of these different species to this mobbing response. And it's great because there's been evidence showing that they all respond to each other's calls. And so it's kind of that big picture question of how is this human noise then going to affect this anti-predator behavior, which importantly, is helping them not get eaten and has some direct consequences for their fitness. So previous work already done in the lab has kind of tested simple approaches. So our lab tends to use a lot of playback studies in which we're playing back recordings we've taken from the fields and kind of measure behavior in response to those recordings. And so what you see here is on the y-axis is basically the mean number of approaches, right? So are they approaching, which is typically what we think of in these anti-predator responses. And then on the x-axis, you see the playback type. So you have a mobbing call on the left, just strict anthropogenic or traffic noise in the middle, and finally a mixed playback that has both the call and the traffic noise. And so it was found, first of all, that chickadees and titmice tend to make the most approaches in general. But you do see that there is this kind of general decrease in their responses when you add noise into the picture. So there is some kind of detrimental effect on their behavior due to traffic noise. But the big question is kind of why is this happening, right? We know that there is this decrease in behavior, but it's still unclear as to kind of the mechanism or the explanation as to why. And so then this kind of big question is what led me to propose to our lab an experiment to kind of figure out which mechanism it is. So several potential hypotheses that could explain these reduced behaviors. The first one is called masking, where basically noise is overlapping the sound of interest and making it harder to hear, right? So this is primarily in the acoustic domain, right? Where basically the sound of the noise is kind of directly overlapping with the sound of that signal, and it makes it hard to hear, or it masks it. The other explanation is potentially distraction, right? So noise can be distracting. Noise in itself may compete for attention with other relevant stimuli. I always like to, the great example with this one is, right, if you're trying to you're driving around, you've got your music on blast, and then you have to go park yourself in a tight spot or your parallel park, and you turn the volume down to try to do it, right? Even though that music doesn't actually have an impact on what you can see or how well you're able to park, right? So noise has the potential to be just distracting in general. And so cool thing about this then is it spans multiple sensory domains, right? It's not just limited to that kind of acoustic of like, oh, this sound mask, the other one, distracting can really distract you from anything. And so the big question is then kind of how can we distinguish between these two, or what's a way that we can get at these two hypotheses? Well, first of all, let me tell you something else cool about chickadees and the chickadee system. So chickadees in their calls, right? So I mentioned they do this chickadee-dee-dee call. They can actually change different elements of their call, so include more or less D notes. So for example, a chickadee-dee-dee-dee-dee-dee call is very different from a chickadee-dee call, right? And they can actually encode predator threat in that number of D notes. So what you're seeing here is basically a study where they presented a bunch of different kind of mounts to these chickadees and measured the amounts of D notes in their calls. And first of all, chickadees, their direct predators are actually these smaller, kind of more maneuverable birds, because those are the birds that can kind of get through the branches where chickadees usually are, and also for whom a chickadee is a much bigger meal, right? So a more dangerous predator for these chickadees are saw-whet owls and pygmy owls, and there's the Cooper's hawk. And basically, the more dangerous a predator is, the more denotes they have in their call. And so because of this, what we can do is kind of utilize this difference in the sensory domains, as well as kind of this idea that you can encode the same kind of predator information, both from a predator itself, as well as kind of a call. And so what we can do is present them either with a visual stimulus. This guy on the left here, affectionately named Raoul, is a taxidermied saw-whet owl. And so we used him as kind of our visual stimulus. And then our acoustic stimulus, we presented then chickadee calls or alarm calls that encode the same level of threat. And the idea is that you can present either one of these in quiet, basically, or you can present them in noise. And so the cool thing is that, right, since masking is only in the acoustic domain, if noise is affecting their behavior through that, then you're only going to see decreased responses to that acoustic stimuli in the presence of noise, but not responses to the visual stimuli, because masking really only occurs in that auditory domain. Versus if distraction is at play, then noise is going to be distracting no matter what the stimulus is, whether it's acoustic or visual. And so we can measure different things, right? We can measure approach responses as well as aspects of their call. And so the great part about this is I at Vassar have access to the Vassar Farm and Ecological Preserve, which was just a 10-minute walk from my main campus. It's about 1,000 acres, so makes up almost half of our campus space. And there's known populations of chickadees, titmice, and nuthatches, and we've had them for years, which is great. And so we have 14 of these wooden platforms that you see on the right that are scattered around the farm that we stock year round with black oil sunflower seed to encourage the birds to come to our platform. And so this video I'm about to play for you is a video that I actually took myself several years ago with a chickadee coming. And so the cool thing is that we can basically use these platforms as kind of a centralized location and have the birds come to us rather than us having to, scramble out in the field and try to find populations of them. That was one of my favorite things about being in the field, is just how close I am able to get to all these birds. So this is just kind of a quick overview of our methods that we use to kind of collect our data over the course of 2021 to 2022. In the middle, you can see our feeding platform. That's basically that platform you had just seen. And so just the right, you can see we either play these chickadee calls or present the stimulus, as well as then for half the trials, then we present them with noise. We have a little recording device to record their calls. And then finally, for like number of approaches, we set up concentric circles of 5, 10, and 20 meters and use 3 observers to basically count those approaches visually of the birds going to and from the platform. And so what did we find? So we're looking at mostly the calling behavior for them. But so for chickadees, we found that their calling behavior was actually decreased to both visual and acoustic stimuli, right? And so which may suggest that this response was modulated by distraction. For nuthatches, we found something different though. They only seem to decrease their calling behavior in response to the acoustic stimuli, but not the visual stimuli, right? So that kind of suggests that their behavior is modulated by masking. And then the titmice showed absolutely no change in their behavioral response, right? Which is kind of like a and so some of the things our lab is thinking about is, well, first of all, I like to say the titmice are always the angriest no matter what. They were always the ones where if you're walking around, they start yelling at you first and all sorts of things. So we like to joke, oh, they're just angry no matter whether there's noise or not. But also another thing our lab was thinking about is the fact that we are kind of using relatively low levels of traffic noise. So it could be that these titmice are kind of not affected by the low levels that we were using. But then again comes the question of why, right? We see this difference in these species, which first of all is pretty cool because it shows that not all animals are behaving in the same way. But then the question is why? Why is this happening? And so this then leads to the fun part of the neuroscience, and we can finally kind of use neuroscience and try to explain these differences based on their auditory processing capabilities, or basically their hearing systems. And not very much is known about these species in particular and their abilities to kind of process signals and noise. So this is what I chose to focus on kind of for my senior thesis. So there's several ways that we can assess hearing or measure hearing. And so, for example, we can try and look at what makes sounds detectable. This is pretty relevant for, mobbing context, because if you're trying to recruit individuals to mob, you want birds from far away to be able to detect your signal pretty well and detect your call. And so the first thing I'll talk about are kind of thresholds, or basically you can think of it as what's the softest sound our birds can hear, which is going to tell us how far away they can hear it from. So how do we do this though, right? What are we even measuring? Unfortunately, you cannot go to a bird and ask it, hey, can you tell me what you're hearing, right? And so instead, we have to kind of use neuroscience techniques to do this. And we use a technique that's commonly used with other organisms that can't talk. which are called auditory evoked potentials. And so basically, similarly, this is similar to the usage with newborn babies, right? Where you're trying to tell if that peripheral auditory system is intact. And so what we're doing is using auditory signals, or tones in our case, to kind of evoke this neuronal response in the form of action potentials, which is kind of a response of a neuron, and measure those potentials to understand how these neurons are firing. And so for my work conducted in the spring of last year, I would go out to the farm, catch wild birds, bring them back, run my experiments with them, and then return them back that same day. So I'm understanding the neural processing of these wild caught birds. And so we use electrodes for them, which you can see on my picture on the right, including one that goes directly over their ear to basically measure that response of the auditory nerve. And so this is kind of a picture of the setup of what our experiments look like for this. And so they take place in an anechoic, or a sound deadening chamber, hence all the black foam padding that you see on the wall. The black sphere that you see kind of at the top of the picture is our speaker, and that's where we play our tones for. And then you can see the bird is down there in the cage, right? And the cage is meant to block out basically the rest of the electrical signals of things like the building and the computers, right? Because what we're trying to measure is an electrical signal of a little bird's little auditory nerve. And so it's important that we kind of get rid of the rest of the electric noise. And so we start by playing them tones from a speaker, and we start playing them really loud, then kind of get softer and softer. And so that point at which their auditory system or their nerve stops detecting the sound is considered their threshold. So I'm about to play some more audio, and I'm actually going to play for you guys what the tones that we are playing for the birds are sounding like. So this is what it sounds like as it's coming out of the speaker. And so part of the reason it sounds like that is because we're playing these tones at about 5 milliseconds per. And so it sounds, really choppy to us. But as you can kind of hear, we slowly step down the levels, starting louder and getting softer and softer. And so, of course, though, our lab is interested then in how noise is affecting these auditory processing capabilities. And so we can do the same thing, but then play white noise in the background at the same time. And so this is what it sounds like when you add noise into the background. And so even just for us with our human ears, right, you can already tell that the noise makes it harder to be able to hear that tone, and that B, as that tone, volume gets smaller and smaller, right, it makes that tone harder and harder to perceive, even if it was relatively easy and quiet. So you can think of these as thresholds, and then when you introduce noise, we call them mask thresholds because there's some sort of masking that's going on. And so we are specifically looking at these auditory brainstem responses, or ABRs, which measure kind of the summed response of the entire auditory nerve to the onset of sound. And so we're looking at the general response of a bunch of neurons. And so the bigger that response is, the bigger that shape is, then the bigger the neuronal response, which means there's a larger number of neurons that are kind of being recruited to respond. And so this is a waveform that I actually collected myself. The red arrow indicates when I started the sound from the speaker. And so you can see it takes a little bit of time for the sound to travel all the way to the bird's ear and then to actually be processed. And so as I mentioned, you can start by playing a tone really loud. So that's what 60 dB is about, is basically the volume of our tone, or the amplitude of our tone. And you can get that quieter and quieter and quieter. And these are all different ABRs in response to that, until you reach a point visually where that response disappears, right? And so this is how we can classify thresholds, or basically what's the point at which our auditory nerve stops responding to these tones. And so this kind of corresponds to then the softest sound they can hear. So with our data, what do we find? So first, what you're looking at is for chickadees. Along the x-axis are different frequencies, because we did this for different frequencies of tones. On the y-axis, you then see their thresholds, right? And so a quieter threshold or a lower threshold indicates better sensitivity, because it means they're able to kind of pick up those softer sounds. And so here's our chickadee data. Here's our titmice data. And finally, here's our nuthatches. And so these are considered audiograms because they're done in quiet. What you'll notice is they all kind of have a pretty similar, a little bit of a U shape almost, right? Where they have this kind of sweet spot right in the middle. This is pretty common for a lot of animals where you tend to have best hearing in a certain range of frequencies. So here's what our three species in quiet, right? This is what they're auditorily processing. And so you can play a low level of white noise in the background. And so at a low level of noise, equivalent to kind of sounds you might hear walking around a nature preserve, you can see that in general, thresholds are increasing, right? Which makes sense because noise makes it harder to hear. But interestingly enough, you see this really large jump for the nuthatches. While in quiet, right, they have the best sensitivity of the lowest thresholds. All of a sudden, you introduce just a little bit of noise, and their thresholds have dramatically increased. The same thing kind of applies with a medium level of noise. So again, this increase in threshold with this disproportionate increase in the nuthatches. And then with a louder level of noise, which is equivalent to basically kind of traffic noise. And so that trend is still continuing. And so basically, just visually looking at this data, you can tell that there's these really clear species differences in how noise is affecting their processing abilities. And so it's really interesting to think about the implications that that might have for the behavior. Another aspect of understanding detection, especially noise, is to understand critical ratios. And so what are critical ratios? Basically, the critical ratio is just how much louder a sound has to be than the level of noise in order to be perceived. So it's pretty easy once you have your mass thresholds to calculate your critical ratios because all you have to do is take the thresholds and then subtract the noise level. And so this is then for titmice, right? What you're looking at is basically you can take this difference, right, from the lowest level of noise between the threshold as well as the masking noise. Here's for the medium level of noise, you take that difference. And then finally, for the loudest level of noise, you take that distance. And so what you'll notice too is that the magnitude of these differences, or basically the length of the arrows, is the same like across a single frequency. It's the same across noise levels. So the idea is that for critical ratios, it doesn't matter how loud your actual noise is, your threshold is always going to be some constant level above that. And so that's what you can then measure as the critical ratio. So kind of similar to thresholds, right? Where smaller thresholds indicate better sensitivities, smaller critical ratios also indicate kind of better sensitivities. And so larger critical ratios just means that sounds need to be even louder than noise in order to be detectable. So similar to our thresholds pattern, right? We see this really interesting pattern where at low frequencies, all the air bars are overlapping. There's no statistical kind of significance in terms of species differing from each other. But once you get to these higher frequencies, you once again see these nuthatches having these really high critical ratios compared to the other two species. And so this is also interesting given that kind of the frequencies that you see this, 2 to 4 range, these are also the frequencies at which their vocalizations have the most energy. So this actually may have some implications for basically the way that noise is going to affect their ability to process the signals of other individuals, both of their own species and then of the other species. And so now we kind of have an understanding of how detection of sounds is affected by noise. But another aspect of hearing that you can look to understand is discrimination. So basically, when sounds are really loud and you can hear them, how well are they being discriminated, kind of, or how are those loud sounds being processed? And so we can measure this by looking at the amplitudes of our ABR responses to pretty loud tones to understand how they respond to these tones that they can definitely hear. And so when we play these sounds well above their thresholds, we get pretty reliable responses. So the tones themselves here are played at 60 dB, which is basically, I think it's about human conversational level. And so the positive peak of that ABR represents kind of the general response of the summed units. We're looking at this, of course, across species. And so for us visually, we can tell, oh yeah, it's a very nice response for all three species. Now you can play this same really loud tone, but then introduce levels of noise, right? So again, kind of a low, an intermediate, and a great level of noise. And once again, you can kind of visually already tell, right? This nuthatch response is going away quite faster than the other two. And then we can actually measure this kind of quantitatively by looking at this voltage difference between the first peak and the second peak. And that's our ABR amplitude that we can then plot. And so again, this is when we're in complete silence, right? You have our frequency on the x-axis, our three different species, the amplitude on the y-axis, where a greater amplitude means a greater response. And it's kind of see, a reverse U shape or an upside down where they have the best amplitude to certain frequencies. And so, right, as you add noise, similarly, right, these amplitudes are decreasing, right, which means that decrease in response. And for nuthatches in particular, too, that neuronal response is getting much smaller, much faster. And so basically all this kind of summed up is basically implying that noise has really big effects on sounds that are even relatively loud, right? As I mentioned, 60 dB is the equivalent of human conversation level. And so because that means that noise is going to have, particularly detrimental effects for these species in terms of their hearing, especially for these nuthatches. So kind of tying all this together, right, I've taken us through communication, behavior, and finally that neuroscience. How does this all link together? And so first of all, kind of the main message that if anything, I want you to take away is that these different species are impacted differently. Not only did our three species show these different behavior responses in our playback experiments, but they also then had differences in their kind of underlying physiological auditory capabilities. And interesting enough with our nuthatches, whose behavior, right, seemed best explained by this masking, right, in that auditory domain, they also seem to have the auditory system that was most susceptible to noise, which kind of leads me to this next point, where these aspects of simple underlying physiology, right, I'm just measuring the response of their auditory nerve, but it may actually help to explain these more complex behaviors. And so the cool part about this is that, if more of these relationships are kind of figured out in the future, I could potentially go collect critical ratios of other species of birds and then be able to make predictions about what their behavior might look like in increasing levels of noise. And so kind of on this note, what are the future directions for this study? You see me there on the right presenting a poster that I made about this thesis data. So not all species are equal, right? But more comparative work is needed, especially in songbirds and with the hearing. As I said, like earlier, the receivers is kind of an understudied side of communication. And so it's really interesting to think about it from that aspect. And similarly, this ABR and AEP technique that I used in my lab has only been done with 3 other species of songbirds. And so there's a lot more, as I said, more comparative work that needs to be done in order to kind of create a more robust framework that we can then make more implications for behavior. Additionally, there's different kinds of techniques that could give us different kinds of results. So the AEPs that we're using are fast and non-invasive, and they're great to kind of collect data as like a first preliminary. But there are other different kinds of techniques. So for example, psychophysical studies in which you can basically train birds to respond by pressing levers and have them respond when they can or cannot hear a tone. Those may be in some ways more realistic in terms of what they're actually behaviorally doing. They're allowed to integrate over time. It's not just the response to their auditory nerve. But on the other hand, it takes, months of training, very small sample size. And so it's not necessarily that one is better than the other, but they can provide us with different kinds of information that may tell us of a bigger picture overall. And finally, this kind of has important implications for both behavior as well as resulting fitness in these increasing levels of anthropogenic noise, right? Human traffic noise is not going to be stopping anytime soon, most likely. And so this is going to have real-time kind of impacts on the way these birds are able to behave, as well as, their survivability. Although there's an interesting study done by someone from UTK, Liz Derryberry, where they actually studied kind of song production before and after COVID, because COVID actually reduced the amounts of traffic noise by quite a lot. And so it's a pretty cool, I think it's a science paper, but they looked at both kind of song production as well as different aspects of kind of communication before and after COVID. So that was pretty cool. But point being, noise has these important kind of impacts on these birds' behavior, which can be important for conservation as well. And so I'd like to give a special thanks to the members of my Gala lab who kind of helped with all the data collection as well as the writing. We did turn it into a manuscript that has been published. It's a lot more statistics and kind of the nitty gritties, but you can go find it if you'd like. And then also Dr. Gall's research website is there on that page. And then finally, thanks to all of you for lending an ear. This is really exciting stuff that I'm thinking about and currently trying to turn my senior thesis data into a manuscript to be published as well. So it's fun stuff, and I always love talking about these birds. So yeah, that's it for me. Yes, now would be a great time to go through some questions.
Trina, that was fantastic. Thank you so much. It was really fascinating. And we do have some questions in the chat. Are you able to see the chat?
Yeah, let me just get it.
I can read it to you as well. So the first question is, why did you make the decision to go loud to soft rather than soft to loud? Wouldn't the loud sound first downregulate the hearing sensitivity?
That's a great question, and both techniques are used in kind of different studies and methodologies. I personally, me and my advisor chose to go loud to soft because it's much easier to see that definitive cutoff between where that threshold kind of is there versus not. Versus if you start soft and get loud, it can be much harder to visually determine where that response actually starts occurring. And so you do run into the things of, yes, that, you start with a really loud sound and it's kind of like, oh, you can't freak out the bird's nerve too much or can't have it firing too much. But there's kind of trade-offs for both ways that you can go. And so for us, we chose to go loud to soft just because we're using kind of a visual determination method where I'm just looking at that, scale of the from loud to soft and visually determining what that cutoff is. And so since that was the way that we were measuring it, we chose to then go with one that's going to make that visual detection easier to see.
Excellent. So let's see. Ellen has a question. Why test with such low frequency? Birds excel at high frequencies, or did you use varied frequencies for test noise?
Yeah, so it's a great question. The biggest thing I'd say is that basically different kinds of birds have different hearing ranges, and that not only is it just across birds, but also subgroups of birds. So there's a huge variation in their what they're able to hear. And so with our songbirds in particular, since we're trying to study how they respond to other songbirds, what we're most interested in is that kind of range of the other songbird kind of vocalizations, as well as for these three species in particular, and most songbirds, it's kind of that 2 to 5 kilohertz range tends to be where they're the best at hearing. And it's kind of like once you go either end off, so I also did like 1 kilohertz and 8 kilohertz, but like you see a dramatic like drop off basically once you get outside of this range of kind of best fit. And so that was something that I was researching as I was trying to figure out, which frequency should I use. And that's how I kind of ended up in this 2 to 4 ranges, just because of these species and the unique situation that they're kind of utilizing. That's what best worked for us. And then the noise that we used was basically white noise. So noise of a bunch of different frequencies that were going to overlap with this.
Excellent. Thank you. Let's see. Yvonne says birds living in the Bay Area are subject to a lot of airplane noise near the airports, but also in places like the Berkeley Hills, given the concentration of flight paths by the next gen FAA. This would be a good project to assess the effect on nesting productivity under these flight paths. So I don't know if you want to respond to that.
Yeah, no, that's definitely a good point, and that's part of what this research is so cool is about thinking about these things, right? Because even though this was about anti-predator response, right, that traffic noise is going to have some sort of other behavioral response that then is going to affect nesting. And another interesting thing too to think about is like which species are better at adapting to these more urban environments than others. So for example, chickadees are just very good at adapting to kind of any situation, and that's why they're so common in a lot of kind of urban areas. But for example, nuthatches are not so much. And so it's kind of one of those things that this arises more questions and more applications, but that would definitely be something cool to think about.
Excellent point. Barbara says, really cool research that may explain why some birds scatter with any human sounds and others remain out in the open. I don't know if you thought about that when you were doing your research.
Yeah, no, that's something I definitely did not think about, right? Because I'm thinking about these kind of more global human noises as opposed to singular sounds. But I mean, species level differences is kind of the key takeaway. And so that would also be something cool to think about.
Excellent. Ori asks, do you have any hypotheses given your physiological data as to why one species shows masking, one showed distractions, and one showed no effect?
Yeah, no, that's a great point. So in terms of masking, for me, my physiological data definitely kind of shows why nuthatches specifically might be impacted by masking, right? They're the most susceptible to noise. And so that kind of makes sense behaviorally. Another cool thing that we were thinking about with our behavioral data is the different roles that these species play in their social flocks, right? So they're forming this mixed flock. But as I said, chickadees and titmice tend to be more of the focal species, where they're the ones who are mostly alerting and are the ones who are kind of on guard and alerting if there's a predator, versus the nuthatches are kind of what's called satellite species, where they kind of like hang along on the outskirts and are listening in. And so in some ways it could be, that the chickadees, right, because they're one of the focal species, they constantly have to keep alert, that they're more susceptible to this distraction because they have to do so many more things. As I mentioned with the titmice, it could be that, our levels of noise, right? I mean, my physiological data kind of showed that the titmice were actually the best at kind of, processing signals and noise. And so if our noise just wasn't loud enough, then it might not have had any effect on their behavior. And so that could kind of be explained by that way. And then finally with those nuthatches, right? Once again, the masking, but then also a funny thing about them, if any of you guys have seen nuthatches climbing around, they tend to go like this on trees, right? So unlike the chickadees that are hopping around and titmice that are hopping around, nuthatches tend to go kind of up and down on tree trunks. And so because of that, in some ways, we think they might be more reliant on their auditory system than their visual, because visual is kind of, you're limited to just what's in front of you / slightly behind you, versus a chickadee who's standing up and kind of moving around. You can use more of that visual space. So it could be that it's because they're more reliant on those auditory cues than the visual cues that make them more susceptible to masking. So there's a lot of different possibilities out here, but definitely physiology is one of the ways that could be explaining this difference in behavior.
Well, just in the examples you shared, I feel like you've added a level of bird watching for all of us to be observing sort of that behavior and wondering about what they're hearing, or are they relying on other senses more? So I know I'm going to be thinking about that as I go out there. Just want to share a kudos from Marie, who says, congrats on your paper. Great data. Thank you. Irene has a question. How did you catch the birds for your experiment?
Yeah, so for the behavioral experiments, we just had the seeds that we would wait for a couple birds to come in the area and then started playback experiments. And then for the neuroscience stuff, what I would do is basically have these cages that we would put on the platforms. And so I'd kind of get them used to the cages first by having the cages with some seed in there and leaving the door open. And it was basically a weight triggered cage. And so I'd get them used to just having the cage there first for like a week. And then the next week I would like actually, put the seed so that they had to go all the way into the cage and couldn't just get it from the outside. And then finally, when it was time to conduct experiments, then I would actually go out there and set the cage. And so the cool thing about this is it doesn't cause the birds any harm. There's really no way for them to hurt themselves. And then once they're in there, I'm watching the cage the entire time. And then I'd have a couple bird bags basically with me, reach into the cages, grab the birds, and then take them with me for the study. The cool thing too is that we never run birds more than once through the neuroscience stuff just to be safe, really. And of course, I released them at kind of the same platform that I caught them on. And so because we only run them once, right, we band all of our birds as well. So we can tell if we've caught one or not. And so especially towards the end of my data collection, after I've been doing this for like a month and a half, I started to catch some of the same birds over and over again. And it was like, okay, well, thank you for coming in my traps, but I need new individuals. But it was also good because it meant that the experience that we were putting them through was clearly not aversive enough for them to, never want to come here again.
Let's see. The next question is from Yvonne. Did you happen to measure the acoustical response in the lab of the 3 species to frequencies of the great horned owl male call? I know these species are only really active during the day.
Yeah, that's a good question. So we did not record their responses to basically the predators themselves. We were just kind of using the chickadee calls to kind of incite the same mobbing behavior. But that would definitely be something cool, would be to use like an actual predator's kind of call itself to see if that will elicit some sort of response. But yeah, we did not use the great horned owl. So that's quite cool.
Well, there's so many next steps that your research lays a baseline for all those new questions that we can ask. Let's see. Ori asks, how did you secure the birds for measuring the nerve signals? Do you have a sense of how stressed they were while secured?
We actually anesthetize them slightly, so just enough that they're not going to feel any of the pain technically associated with this, even though it's not really that much pain. But all we have to go through kind of IACUC protocols. So for those of you who haven't heard of that, which is probably most, is like the Institutional Animal Care and Use Committee. And so every single school or research institution has them, where basically before you do any sort of research, you have to say, here's what I'm proposing to do. And one of the things that they always talk about is minimizing animal harm. And so most of the time, a lot of the data that we're going off of for our anesthetation or the things that we're using is based off of previous studies that have shown that this doesn't cause any harm, this doesn't cause any stress kind of thing. So yeah, so we anesthetize them basically to the point where, they're not going to feel the pain, but we can still get that, physiological response. And so it's a very tight balancing act because of course we care for the birds, right? We want to make sure that nothing bad happens to them. And so we have a lot of things in procedure for that. So for example, we're keeping their bodies at a certain temperature the whole time, and we're monitoring that very carefully. And, if the bird starts to wake up, we immediately stop our studies. And so that was kind of what got in the way of a couple of my individuals, for example, is they started to wake up, or the mix of, the drugs that we were using wasn't working quite right. And so the most important thing was just stopping the study and making sure the bird was okay. And then once we go through those, then what we do is then we put them kind of in the cage that we're keeping them at on a heat pad. And then with a towel, we rest them, and basically we wait for them to fully wake up and to fully start eating before we release them back in the wild. So we have a lot of kind of procedures in place to make sure that they are well cared for and that they're okay. And always the bird's safety and health is kind of the priority, especially since these are wild caught birds, right? They're not from the lab. They're not raised in labs. They are from the forest. And we want to make sure those populations continue to do well.
So a very careful protocol. Thank you for sharing all that. I know it's super interesting. Joanne asks, what other songbirds have been tested?
Yeah, so the birds in general that tend to be kind of have the most data on them are typically budgergars and canaries, as well as zebra finches. Zebra finches are kind of the classic when you think of laboratory birds and studying things like hormones and stuff. But yeah, in terms of, for example, the AEP data, it's mostly kind of, I think, budgergars. And great tits are another one. They're kind of related to the black-capped chickadees. Carolina chickadees is another chickadee species. There have been some work in as some like dark-eyed juncos, which I think someone saw as their check-in question. But so there is like kind of a small pool of songbirds in which this stuff has been studied in, which is pretty cool. But not as much with the aspect of noise, right? It's a lot about just the general hearing, but not so much the hearing and noise part about it. So.
Okay, excellent. Let's see, Ori says, sounds like the procedures were well thought out. Would the anesthesia affect their ability to detect the sounds?
Yeah, so in fact it did, right? Because what we're giving them is kind of a combination of a muscle relaxant as well as kind of a sedative. And so if you give too much of part, like, that one part of drug, it actually causes the response to not be there as much. And so I would have to wait basically until they're in kind of that perfect state where what we do is kind of in between playing these tones, we just play them clicks what we call clicks that are basically meant to elucidate like a really big response. And I would play clicks between all these tones to make sure they're responding kind of as I expect. And so I'd look for this general amplitude that's within. But as I said, it's a very stressful process where I kind of have a very small window to do all this data collection. But yes, the anesthesia does actually affect. And so sometimes if we administer, a certain amount of the drug that we calculate based on the bird's body weight, and sometimes it still is, too much for the bird. And so then they go under really deep really quickly. And so then I'm not getting any real elicitable responses. So I basically have to wait then until they wake up a little bit more in order to then actually collect the data that I need to. So
when it's almost like that in of itself is good research and information for people who want to do these kind of studies to look at those variables and lots of moving parts as you're doing all that for sure.
yeah.
Dudley says, have there been any studies on the noisier birds, for example, jays and crows, and their response to anthropogenic noise?
Yeah, that's a great question. I don't know off the top of my head, since kind of most of the literature that I delve in is these songbirds, but I'm sure there might be somewhere out there, especially because anthropogenic noise just in general is starting to become more of a topic of interest too. I recently helped collaborate on kind of a review paper that looked at a bunch of kind of thinking about anthropogenic noise and how that affects the hearing of many different species, not just songbirds. So I don't know if there's anything specifically for those species, but there has been kind of an increasing interest in the effect of anthropogenic noise on a bunch of different birds. So yeah, I don't know off the top of my head, but I'm sure there's something out there, honestly.
Does anybody else have any questions? I had a couple of thoughts, but if you have more questions, go ahead and put it in the chat. I feel like listening to your talk, I know we had a talk a few years ago about soundscapes, and it was an introduction for me about going in forest and how you can measure the health of an entire ecosystem by the sound itself. And then you've kind of narrowed down, a kind of different question around what birds hear, but I can see lots of applications that organizations like SFBVO can use in the future in terms of promoting the sustainability of these species and looking at the impacts that we're having, more subtle impacts. We know about habitat loss, but this is a different kind of habitat loss, which I think just opens so many potential studies and also work with land managers and conservationists to make changes and to be specifically looking at how sound is affecting birds, which I think a lot of people we're so worried about what's going on with just their habitats that we don't necessarily know these other things are part of their habitat. Ori says, what are you going to focus on in your PhD?
Yeah. So funny enough, this kind of leads into what I was going to respond to you with too, where so my interest is in the communication itself and these signals and how these signals evolve. And so for me, it's really interesting to think of this shift in soundscape, right? Because these signals have evolved in already these kinds of environments that are pretty complex. And then all of a sudden you throw noise in there. And so then the question is kind of how are those signals evolving being more in response to that? So that's kind of the basis for my PhD work is with frogs. You can think of it in the context of sexual selection. And so females are choosing the males that they want to mate with. And so a lot of my questions kind of ask about the male signaling side of things, how females are responding to that. Particularly for now, the cool thing about signaling, right, is that it's never constant, right? So it's not like we're all robots and we produce signals that are exactly the same every single time. So you actually get this inherent kind of what we call within individual variability, or this variation in the way that calls are produced, not only by birds, but especially by frogs. And so my questions right now are kind of asking, do females and receivers actually attend to this information? Is this something that they kind of can pick up on, and if so, make decisions about it? And so still very much on the receiver and the decision-making side of things, but just with like a slightly different kind of topic of animal and in a different context. But it's always cool because animal research, right? I just have this big question about, how these signals are affected by their contexts and their environments that they're in. And so I can use lots of different kind of study systems in order to answer these questions.
Joanne asks, why did you go away from bird research? We've got a lot of bird lovers here, so.
Yeah, no, that's a great question. It mainly had to do with kind of finding the right lab that was going to be able to answer the questions that I want to. And so, of course, there are more labs. And so, for example, at UTK, Todd Freeberg's lab looks at kind of these mixed species of these three same species and the way that they kind of have behaviors. But since I was interested in kind of combining my neuroscience and behavioral research, it led me to finding my current lab advisor, Jessie, who just started her position last year. And so she's asking a lot of really exciting questions kind of about this communication system. And I think there's a lot of really cool things to ask about in frogs. Not that you can't ask them in birds, but it just kind of offers a different way to explore them, especially with males. They have these really consistent calls. It's much easier to do call analysis with male frogs than it is for songbirds, because songbirds like kind of calls are much more variable, and so you can only really classify them as note type. So like the A, B, C, D, and of course you can talk about things like, rate and how often they're calling. But it's way more kind of, you can analyze a lot more with frog calls. So they have calls, they have pulses within these calls. Their call effort and duration is directly linked to kind of their physical fitness. And so in some ways, females can kind of make some decisions about, oh, this guy has a lower frequency because he's a bigger body and therefore he has a higher fitness. So there's just a lot of interesting questions that you can ask with a frog system in the context of sexual selection. So not that I don't love birds, because I do love birds and I never thought I would be a bird person, but here I am now. But my current focus is kind of shifted to look at frogs. And actually in our lab, we may even be looking at crickets too. It's kind of using this comparative lens where crickets and frogs have evolved to have very similar signals, but they're two completely different species. So the question is kind of, how the same method of communication evolved in these two very different animals that have two different hearing systems as well. So I guess I'll say that a lot of my work loves looking at like comparative work and looking across species and then even across different kinds of animals to understand these bigger questions.
Absolutely. And I was going to say that the fact that you're looking at it with different species actually elevates the bigger question of what does sound have, what the effects sound has on wildlife, and how and what are we doing, right? And so seeing it in all these different kinds of species, the same kind of issues is actually a good conservation question. Dudley says this raises the whole question of light and how it impacts birds. Lots of bits in news. It would be fascinating to hear from someone who's specifically studying this in the way Trina studies communication. Thank you to everyone who participated, and this will be on our website for people to listen to again. I know I want to watch it again and absorb even more of it. But I'm wondering, with Dudley's question, have you interacted with other scientists who are asking questions around other kinds of disturbances like light disturbances, and what are you seeing in common or different?
Yeah, no, light pollution is definitely something that's been thought about. I don't know if it's been discussed in the context of songbirds specifically, but I know that there have been studies on light pollution affecting things like visually signaling animals. And so it definitely is, just like you can kind of think of what I study as like noise pollution, right? Light pollution is its own source of something that has changed in the recent years. And so that definitely is a topic of interest for different researchers, although I don't know off any at the top of my head, but I've definitely read a couple, like one or two papers about light pollution and the impacts that it may have on animal behavior as well. So it's definitely something of interest to people in the field.
Absolutely. And Doug says, I'm so taken with your enthusiasm and engagement. It's very inspiring. We've had a lot of really positive comments and interspersed with the questions as well about the work that you're doing, congratulating you and thanking you for doing this research. It's really important to all of us who are interested in protecting birds and their habitats and other species. And I'm just really excited to find out what's next with this line of questioning and to see, I know you are coming out with you're going to publish this work and it's going to be dispersed to other scientists in that way. Hopefully you'll do other talks like you did for us. There's going to be a lot of graduate and undergraduate students who are going to be interested in taking where you started and kind of expanding on that research. And I can see lots and lots of environmental organizations like SFBBO interested in what you've learned as part of our storytelling when we talk about the importance of conservation and the many different ways we need to think about conservation. Is there anything else you'd like to share to kind of wrap up and your basic conclusions or enthusiasms or next things you want us as a community to be thinking about when we're thinking about birds and hearing and sound?
Yeah, no, I think it's basically the main message is that you can combine a lot of different information from a lot of different things, right? Whether it's just, going out in the field and just enjoying being a bird watcher, right? as well as then, taking a more kind of research side of things, that it all comes together for this one bigger picture of kind of understanding these animals and what they're doing and why they're doing, just because we have an appreciation for what they're doing. And so I think that's where kind of my love and my passion for this research comes from, is basically being like, these animals are so cool, birds are so cool, like I want to understand how they're doing the things they're doing. And so it just requires a lot of different perspectives to kind of get that bigger picture, because of course it all ties in one way or another. So
wonderful. And I heartily agree with you in our conservation message that we want people to value birds, have a curiosity about birds, and connect with what's common with us and birds. And I hope this will elevate your birding and allow you to see birds even with more interesting contexts as you go out and do your birding. And thanks everyone for supporting SFBBO.
SFBBO is a nonprofit. With your support, we will continue to inform decision makers, inspire people to act, and involve diverse communities in our work. Please visit our website to learn more and get involved today.